Phylum Mollusca. Mollusk is a Latin word which means soft. The defining characters of phylum Mollusca are: number 1, the dorsal epithelium forming a mantle which secretes calcareous spicules or one or more shells. Cuticular band of teeth, which is known as radula in the esophagus, is used for feeding, and it is not present in the bivalves. It means that the bivalves are an exception to this character. Ventral body wall muscles develop into locomotory or a clinging foot. Phylum Mollusca is an enormous phylum including about 100,000 described living species, along with another approximately 7,000 species that are known only as fossils. These species are distributed among some extremely dissimilar looking organisms, making the molluscan body plan probably the most malleable in the animal kingdom. As you can see in the pictures, that the clams, snails and octopuses, they are looking very dissimilar, but they all belong to phylum Mollusca. There really is no typical mollusk. Most but not all mollusks have shells consisting primarily of calcium carbonate set in a protein matrix. Organic material may comprise about 35% of the shell dry weight in some gastropod species and up to about 70% of the dry weight of the shell in bivalves. The shells of most mollusks, including gastropods and bivalves, have a thin outer organic layer which is known as the periostracum. Then another thin innermost calcareous layer is known as the nacreous layer, and the middle layer is also calcareous, but this layer is thick and this is known as prismatic layer. The microstructure of shells among the members of different molluscan groups vary from each other. This means that the microstructure of shells also varies from group to group or from class to class. Both the organic and inorganic components of the shell are secreted by specialized tissues known as the mantle. If a grain of sand, parasite, or other foreign particles become trapped between the mantle and the shell's inner surface, a pearl may form over a period of years. Natural pearl formation is a fairly rare event. Perhaps only one oyster in 1,000 will harbor a valuable pearl naturally. The human intervention increases the frequency of pearl production by surgically implanting pieces of shells, which are usually from freshwater bivalves, or plastic spheres between the shells and mantles of mature oysters, and then keeping the oysters alive for another 5 to 7 years. And in these 5 to 7 years, a pearl is formed in a natural way, but with a human intervention. The cultured sperms thus formed as a result of human intervention are in a perfectly normal manner. Humans intervene only in getting the process started. Although the mantle is a major molluscan characteristic, its role varies substantially in different molluscan groups. Similarly, the molluscan foot is also highly modified for a variety of functions in different groups. Most mollusks have a characteristic cavity lying between the mantle and the viscera. This mantle cavity usually houses the comb-like molluscan gills known as ctenidia. Ctenidia is a Greek word which means comb. It also generally serves as the exit site for the excretory, digestive, and reproductive systems. A ctenidium, which is the singular form of ctenidia, when present may have purely respiratory function or may also function in the collection and sorting of food particles. A chemoreceptor or a tactile receptor, which is known as the osphradium, osphroi is a Greek word which means smell, is generally located adjacent to the ctenidium. With the exception of squid and other cephalopods, mollusk gills works on the principle of countercurrent exchange, a system that greatly increases the efficiency of gas exchange between blood flowing within the ctenidial filaments and the water flowing over them. In this system, blood and water flow in opposite directions. This is the micrograph of osphradium. From the mantle cavity of southern oysterel drill, the leaflets of the osphradium, which is 4 to 5 millimeter long in this species, hang down into the in-current water stream within the mantle cavity. Water thus contacts the osphradium before contacting the gill. This figure shows the concurrent exchange in the molluscan gill. As you can see, the direction of water flow across the surface of each ctenidial leaflet is opposite to the direction of blood flow through the gill capillaries. In this figure, as you can see, the efferent vessel carries deoxygenated blood to the ctenidial leaflets from the tissues. Oxygenated blood leaves the gill through the efferent vessel, carrying the blood to the heart and thence to the tissues. The illustrations below are showing the principle of counter-current exchange. Length of the vertical black arrows signifies magnitude of the concentration gradient for oxygen between water and blood. The counter current situation, equilibrium is never attained. Oxygen diffuses along the entire gill sheet surface, permitting a greater percentage of oxygen in the water to be taken up by the blood. Oxygen diffusion in a non-concurrent situation, the oxygen concentration gradient diminishes rapidly, so that beyond point 2 on the gill, very little further diffusion of oxygen take place in case of the current exchange. So the difference between the current and the counter current exchange is that in the counter current exchange, equilibrium is not attained very quickly, while in case of the current exchange, equilibrium is attained and then there is no further diffusion. So in this way, the counter current exchange is a much efficient system. Follow the path of water flow from point 1 to point 3 in the figure. As the water moves from left to right in the diagram, it is always in contact with blood of lower oxygen concentration, maintaining a large concentration gradient for oxygen. Conversely, following the path of blood flow from point 3 to point 1 in the same figure, The blood, even though it is continuously acquiring oxygen from the water, is always coming into contact with water of higher oxygen concentration as it moves from right to left in the figure. Thus, oxygen will diffuse from water to blood as the blood moves over the entire length of the gill filament. In the alternative hypothetical situation, in which water and blood move in the same direction, for reference, see the last figure at the bottom. The magnitude of the oxygen concentration gradient between the water and the blood continually decreases along the surface of the gill sheet as the water loses oxygen to the adjacent blood. The rate of gas exchange between water and blood will decrease correspondingly by 0.2. In this figure, for example, the water has already given up much of its oxygen to the adjacent blood and is nearly at the same oxygen concentration as the blood next to it. Little further, gas exchange between the two fluids will occur as the blood continues to move within the gill past 0.2. In terms of oxygen exchange, the gill surface beyond 0.2 is in effect wasted. The molluscan coelom is very small, being restricted largely to the area surrounding the head and gonads. Some zoologists have suggested that this cavity is in fact not homologous with the conspicuous coelom cavities of annelids and other non-controversial coelomates, and that molluscs evolved directly from acoelomate flatworm ancestors. Current molecular data, however, support an alternative view that molluscs have descended instead from some coelomates ancestors, and that the body cavity experienced a substantial reduction in size in the course of subsequent evolution. Either way, the molluscan coelom is small and has no locomotory role. On the other hand, blood sinuses comprising the hemocoel blood cavity are well developed. This hemocoel serves as hydrostatic skeleton in locomotion of some molluscs. Many molluscs possess a feeding structure known as the radula. The radula consists of a firm ribbon composed of chitin and protein, along which are found numerous rows of sharp chitinous teeth. The ribbon is produced from a radular sac and is underlain by a supportive cartilage-like structure called the odontophore. The odontophore radular assembly, together with its complex musculature, is known as the buccal mass or the odontophore complex. For feeding, the buccal mass is protracted so that the odontophore extends just beyond the mouth. The radular ribbon is then moved forward over the leading edge of the supporting odontophore and then pulled back. As each row of teeth passes back over the edge of the odontophore, the teeth automatically stand upright and rotate laterally, rasping food particles from the substrate and bringing them into the mouth as the radula is withdrawn. As old teeth are worn down or broken off at the anterior end of the radular ribbon, new teeth are continuously being formed and added onto the ribbon's posterior end in the radular sac. This is the longitudinal section through the anterior of a gastropod, showing the relationship between the odontophore, radular ribbon, and mouth. This structure shows the anterior end of a moon snail, which is a Euspira species, with the radula visible in the mouth opening. The accessory boring organ secretes acid and enzymes that allow these carnivores to bore through the shells of other molluscs. The cautious wording about mollusks are that generalizations about mollusks are difficult to make. For example, adult mollusks cover an enormous size range, from a shell length of less than 1 to 2 millimeters in some marine gastropods to body lengths exceeding 12 meters for some squids. Extant mollusks are distributed among 7 classes. 6 of these 7 classes are represented by fossils that formed some 450 million years ago, along with one additional class of mollusks, the Rostroconchia, whose clam-like members went extinct some 225 million years ago. Only one class of mollusks, the Aplacophora, has apparently left no fossil record. The evolutionary relationship among the different molluscan groups have been argued about for over 100 years, and the arguments have not yet ended. The fight over molluscan evolution may even now just be warming up, but at least we have a clearer idea of what the battlefield now looks like.